We're going to move on to our third talk, which is going to be presented by Dr. Jed Lampe. He is a multi-R01 funded investigator, educator, and scientific leader in ADME and PK. He received his PhD at University of Washington and his postdoc at UCSF. Now he's an associate professor at the University of Colorado Skaggs School of Pharmacy. His research interests include the application of bioanalytical, biophysical, and computational techniques to understand and predict drug metabolism and toxicity in special groups, particularly neonates and in pregnancy. He's made seminal contributions to our understanding of drug metabolism and toxicity, particularly in the developing fetus and the neonate. He serves as the chair of the NIH DBDT study section, as well as the FDA Scientific Advisory Board, and on multiple editorial journals. Welcome, Jed.
Okay, great. It's great to be here. I just want to say a quick thank you to Lauren for inviting me to be part of the session, and then the organizers in general. It's been a great meeting so far. I look forward to the rest of it. So today I'm going to talk to you about our work with iPSC cells, trying to differentiate them to produce a neonatal hepatocyte-like cell. But first I want to talk to you about babies. They're strange creatures, aren't they? Very, very cute, like this one, but strange. You know, looks like she's munching on her own foot there. Very odd behavior. But they look a lot like us, right, as adults on the outside. But you scratch the surface, you look under the hood, and they're very different. Their physiology, anatomy is very different. It's a moving target. It's continually changing. So why is this important? Well, my lab studies drug metabolism and disposition, as Lauren was saying. And I always ask this question because it's something that keeps me up at night, right? Can we treat babies just like little adults when it comes to metabolism and disposition? Now, normally I'd spend 2 or 3 minutes on this slide trying to convince the audience that the answer was no, but you already know that, so we can move right along, right? The other thing I'd like you to appreciate, though, is that it's not just that babies are not little adults, but it's a moving target, right? So in other words, a fetus is very different than a 1-day-old, is very different than a 1-week-old, 1-month-old, and so on, even though they're all neonates. So why is this important? Well, there's all these age-related changes in anatomy and physiology that affect pharmacokinetics and toxicity. Changes can occur in absorption, tissue distribution, protein binding, blood-brain barrier, renal function, body surface area, liver function, and the one that we really care about in my lab, so that's drug-metabolizing enzymes and transporter isoform expression, right? So why is this important? Well, off-label use of medications in pediatric patients is increasing. Recent meta-study found that about 10% of patients in the NICU are receiving at least one off-label drug, right? All these drugs are given to them off-label. And in many cases, they receive more than one. So polypharmacy is really common in this population group. This increases the risk for drug-drug interactions, and the American Academy of Pediatrics predicts that this is only going to increase in the future. Particularly with pregnant persons, fetuses, and neonates, they're really susceptible to these adverse drug reactions because some medications, such as antiepileptics, antidepressants, and antivirals, can't be discontinued during pregnancy, right? So the risk for fetal exposure is pretty high. And again, polypharmacy is really common among these groups. Now, a few years ago, Ron Hines, I took this table that he produced from Clausen's Casseret and Duell's toxicology textbook, and what he nicely showed in this table here, maybe I can use the mouse, see pointer options, there we go. All right, so he distributed the drug metabolizing enzymes into 3 different classes. Class 1 are the ones that are only expressed during that fetal neonatal period. Class 2 are only expressed are expressed both during the neonatal period and the adult period, and then class 3 is only expressed during the adult period. Now, what jumps out at you is that these are very different, right? I mean, these are very different periods of life where you're going to see dramatic changes in drug expression of drug metabolizing enzymes. And in my lab, we're really focused on CYP3A7 because the CYP3A enzymes, as most of you are aware, are the main pathway for drug metabolism and disposition in both adults and neonates. In adults, you have CYP3A4, it metabolizes more than 50 to 60% of the small molecule drugs in the market. Neonates developing infants have virtually no CYP3A4. They have CYP3A7. Now, these enzymes are pretty similar. There's about 87% sequence identity between the two enzymes, but operationally, functionally, they're very different. CYP3A7 metabolizes a lot of the same substrates, but the KCAT, the turnover rate, is about 1/100th that of CYP3A4. So if you do an allometric scaling, you just scale down for size and weight, you could still be overdosing your infant by 100-fold. And this is kind of pointed out here in this really nice study. It was done some time ago by Jeff Stevens, where he used immunoquantitation techniques to look at a panel of fetal donor livers and neonatal donor livers. And what you can see, which jumps right at you, is that CYP3A7 is very highly expressed in that pre-neonatal phase in the neonatal phase out until about 6 months. And then CYP3A4 expression begins to take over and you start to see that mature phenotype. So again, this is a moving target, right? If you want to use PK models to try to predict drug metabolism disposition in these population groups, you have to realize that that ontogeny is continually changing over time. So this is kind of to illustrate the point here why this is so important, and you can't just treat babies like little adults. There's a study we published a few years ago with this antiviral drug cocktail called Caletra, which is a protease inhibitor. Well, actually, lopinavir is the protease inhibitor. Ritonavir is a pharmacoenhancer, right? So ritonavir is just put in this cocktail to inhibit CYP3A4, so you don't get too fast clearance of lopinavir, so you can maintain relevant physiological levels of the protease inhibitor. I should point out at this point that CYP3A7 also has an important endogenous function, and that's to convert DHEAS into 16-alpha hydroxy DHEAS. And this function is really important to developing fetus and neonate because this is where all the downstream androgens and estrogens come from. If we can't do that first metabolic step, you're going to have adrenal insufficiency, low birth weight, and premature birth, which is exactly what they saw in a cohort of infants that were born to mothers that were on Caletra. If you look here on the left, what we're looking at is some clearance studies that we did looking at the clearance rate of CYP3A4, recombinant CYP3A4, and recombinant CYP3A7 in the presence and absence of ritonavir. Now, if you don't have ritonavir there for CYP3A4, lopinavir, the active drug, gets cleared very fast. So you absolutely need it. But when you have leave ritonavir out of the cocktail for CYP3A7, you see almost no change in the clearance rate. So it really has this extra inhibition component which causes this metabolic liability, toxicity that's associated with the drug that's not necessary for the infants, right? And that was recapitulated here with neonatal human liver microsomes. So truly, you can't treat babies like little adults. But the problem is we don't have very good systems to test drug metabolism for the neonate, right? If we want to do drug metabolism for adults, we can just order hepatocytes, we can order human liver microsomes, very precious resource for neonates because we have low neonatal mortality in this country, which is a good thing, but it makes it hard to study. So my student, fourth-year graduate student in the lab, Emily Gracie, did all the work today that I'm going to show you. She deserves all the credit. She was looking for a model system to try to study drug metabolism toxicity. Of course, again, in adults, human primary hepatocytes are often the gold standard model. We can't get those for neonates. So she started looking at the possibility of developing an iPSC cell model system. So our goal initially was to characterize our sustainably sourced stem cell-derived fetal/neonatal, and I'll go back and forth, they're called FNHLCs or HHLCs. This is the same cell line, but basically hepatocyte-like cells that are derived from iPSC stem cells. So our goal was to characterize these and compare them to neonatal and adult primary human hepatocytes in terms of levels of gene expression, but also enzymatic activity, right? So these functional studies, I believe, are really critically important. It's nice to see genes go up and down, but sometimes those don't always correlate with activity, particularly with P450 enzymes. So it's important to do these functional studies. And we also wanted to demonstrate the utility of the system through xenobiotic metabolism and bioactivation of toxicants. So there's a lot of protocols out there. We weren't the first ones to do this, right? So which one do you choose? We carefully, or actually Emily, carefully looked at all the protocols and decided that there was a little good in each one of them. And so she combined a lot of the steps through trial and error, added some of our own in a manuscript that should be published soon in Drug Metabolism Disposition. This is under review, and it's all detailed there. Or you can ask me for a preprint. Happy to share that information with you if you're interested. Not to belabor the point in terms of our experimental protocols here, but our workflow, but the idea was we did our differentiation step, then we tested the cells in terms of gene expression profiling, compared them again to the adult hepatocytes and the neonatal hepatocytes, and then we also did the metabolic studies to show that we could actually see drug metabolism and metabolism of some of these endogenous metabolites. So let's dive into the data a little bit. So here's our differentiation optimization of these hepatocyte-like cells. You can see here we have pretty high levels of expression of alpha-fetoprotein, low levels of 3A4, higher levels of CYP3A7, and importantly, after optimization, she saw high levels of CYP3A7 metabolic activity. The take-home messages here are that CYP3A7 expression seems to be more regulated by glucocorticoids than PXR activators, and that's been reported in the literature before, so we were happy that we saw that in our cell line as well. Dexamethasone seemed to be the best inducer, so we used that at about a concentration of 100 nanomolar in the differentiation process. And the ideal differentiation length is shown down here at the bottom. Was between 26 and 30 days where we saw maximal CYP3A7 expression and activity. If we take a deeper dive in the gene expression profiling, I won't present all the data here. I just wanted to focus on a subset of genes, 25 plus genes that we looked at. These are involved in phase 1 and phase 2 metabolism, since we were interested in creating a metabolism model. Some fetal specific hepatocyte markers, transcription factors, and transporters. And in general, what we saw was the hepatocyte-like cells really displayed a mix of fetal / neonatal markers and were quite distinct from the adult primary human hepatocytes. So they weren't exactly like neonatal hepatocytes, but they were a lot closer to them metabolically than the adults. And you can see that here in the panels B and C, where they're color-coded, red being more expression and blue being less. If you just take an example of CYP1A2 here, our expression profiles is somewhere kind of in between the adult and the neonate, but a little closer to the neonatal hepatocytes. If you look at the panels on the right, again, CYP3A7 was our marker for activity and success. And so we see a pretty good increase in expression of CYP3A7. It's a lot lower still than the newborn or neonatal hepatocytes here shown in green. We were just able to see activity with 3A7. Actually, sorry, that's a different slide. CYP3A4, however, had much lower expression profile. So that's exactly what we wanted, right? Because then they look a lot more like the newborn hepatocytes. We also saw an increase in alpha-fetoprotein and GST-pi, which are also markers of those neonatal hepatocytes. So, great. We saw these gene expression profile changes. What about activity? As I mentioned earlier, CYP3A7 metabolizes both DHEAS and DHEA to the 16-alpha-hydroxy DHEA. It's an indicative reaction for this particular enzyme. We looked at 2 differentiated cell lines, one male and one female, and compared them both to the neonatal and the adult hepatocytes, primary hepatocytes. And we saw definite activity, signs of activity with both the male and female hepatocytes, indicating that these HLC cells were capable of metabolizing endogenous steroid hormones. Interestingly, now this is an N of 1, so I don't want to make too much out of it, but we did see somewhat of a difference between the male and female cell lines, and with the female cell line expressing a little bit more CYP3A7, having more activity than the male cell line. We also wanted to look at a real drug, right? So great, we can see metabolism as an endogenous marker, but what about a drug substrate? Because that's what we'd like to look at in the future. So we chose a drug, midazolam. This is a well-known CYP3A substrate. Midazolam's metabolized by both CYP3A7 and CYP3A4 to produce 2 different metabolites, the 1'-hydroxy midazolam and the 4'-hydroxy midazolam. Now, as it turns out, CYP3A4 really likes to make the 1-hydroxymidazolam, not so much the 4-hydroxy, right? Whereas CYP3A7 makes about equal amounts of each. So this gave us a really good handle to look at metabolism specifically by CYP3A7 on the background of CYP3A4 by just comparing the ratios of the two metabolites. So you see that over here in the graph to the right. So in the primary human hepatocytes from the adult, you see a lot more 1'-hydroxymidazolam than the 4-hydroxy. In the neonatal cells, it's a lot closer to 1 to 2. And then both our hepatocyte-like, the male and female cells, are much more closely resemble that neonatal ratio of midazolam metabolites, indicating that, yeah, we have a lot more 3A7 present than 3A4. So finally, we wanted to look at bioactivation in this model as well, because we wanted to use it as a model for hepatotoxicants in the neonate and developing fetus. And so we chose this aflatoxin B1. It's a very commonly known compound that's produced by fungus that's actually activated by CYP3A and causes hepatotoxicity through this endo-exoepoxide that's formed here. So we were able to show that we could form both that endo-epoxide and the AFQ1 detoxification product that's shown here in the LC trace on the right. We saw metabolism both in the neonatal hepatocytes and our cells and of course the adults. It seemed to be a little less toxic, like the last speaker was talking about. The compound, I think it was cadmium he was looking at, seemed to be a little less toxic in his model cell system. So we saw that here too, is that we still saw toxicity with aflatoxin, but you had to get to higher concentrations of the primary hepatocytes. I don't know what that means exactly. I think there may be some issues involved with transport there that we want to look at. All right, so just to sum it up, I don't want to take too much time from Ed. These hepatocyte-like 3A7 cells, we believe, are good models to study newborn drug metabolism, disposition, and toxicity. We're still trying to improve the model. As I tell my students, you know, all models are bad, but some are useful, and so this one falls into that category. We have shown that it's optimized for 3A7 expression and activity. Our gene expression profile is closer to the newborn hepatocytes than the adult hepatocytes, so that's good. We also showed, importantly, we showed functional CYP3A7 activity with multiple substrates, both endogenous and a drug substrate there as well. And finally, CYP3A7 mediated induction, I didn't show you that for lack of time, but we are also able to show bioactivation and how that occurs with this particular cell line. All right, so just to wrap it up, I just want to thank everybody in the lab, particularly Emily in the center of the picture here. She did all the work. It was one of those projects that she came to me and said she wanted to do this. I said, I don't think that's going to work. You shouldn't do it. And then she came back again, and I said, all right, try it. And sure enough, she got it to work after about 6 months of trial and error. I'd like to also thank our NIH support and the generous support of the American taxpayer for all our work. Thanks.
Thank you very much, Jed.